Good evening, and thank you for attending our webinar dubbed The COVID-19 Vaccine, Is It Effective? This webinar is part of a series of webinars with the distinguished Dr. Nicole Ramlachan entitled Hype or Hope, Investigating the COVID-19 Vaccines, and it is being hosted by the University of Trinidad and Tobago under the auspices of the Office of the Vice President of Research, Academic and Student Affairs. I am Dr. Samantha Glasgow, Associate Professional in the Health Sciences Unit of the UTT, and I will be your moderator for this evening's proceedings. The agenda will be as follows. We will begin with the main presentation by Dr. Nicole Ramlachan, followed by a question and answer segment where you, the viewership, will have the opportunity to have your questions and concerns addressed by the speaker. You will use the comment field on YouTube to do so. Finally, I will provide information on the upcoming webinars carded for next week and offer closing remarks. Without further ado, I will now introduce our presenter. Dr. Nicole Ramlachan holds a PhD in genetics from Texas A&M University. She has extensive experience in genetic research and is involved in genetic testing locally, including SARS-CoV-2 testing and research. She's also had tissue culture experience and has worked on virology and microbiology projects with research models studying bacterial and HIV retroviral infection, as well as in the production of monoclonal antibodies. Dr. Ramlachan has spent the last 25 years in academia and has worked as a consultant for private companies, as well as for the US and Canadian federal governments and international agencies based in Central and South America. Dr. Ramlachan, an Associate Professor of Biotechnology of the UTT, is also well published internationally. Ladies and gentlemen of our viewing audience, I present Dr. Nicole Ramlachan.
Thank you so much, Dr. Glasgow, for that lovely introduction, and welcome everyone. I'm glad that we are able to come back together today for our second session. And I think this is going to be a little bit of the meatier one that everybody is chomping at the bit for. In terms of the hype or hope story, specifically the COVID-19 vaccines, what are they, how are they made, are they effective? I'd like to start first by going backwards a little bit. We talked about the variants and so on in the first session, but we didn't really discuss what a virus is and what the concept of viral infection. So viruses are actually non-living particles. They are much smaller than bacteria, and they either have DNA or RNA, right? And they either have a protein coat that's a capsid, sometimes they're enveloped, sometimes they've been evolved to have little extra things to make them extra protective against our immune system and so on. The key thing is they cannot replicate by themselves. They need to have a host, and that's where we come in. And the examples of many different viruses that have been able to be prevented via vaccines, things like measles, mumps, rubella, influenza, hepatitis A and B, which are in our normal vaccine schedule since we are about 3 months old. Everybody, alive in the world today probably has had some level of vaccination if they have access to healthcare and healthcare systems. Influenza viruses, which are RNA viruses, are an example of a virus that mutates all the time because of the antigenic nature of the virus. We talked about that a little bit about that on Wednesday. And that is one of the reasons why we have to have a different flu vaccine every year, because we have to kind of predict what's going to be in the future and then make our vaccines to suit. Now, what is great about SARS-CoV-2 vaccines is that SARS-CoV-2 is an RNA virus, and we've been able to actually utilize that against the virus in developing the vaccines, right? So the COVID-19 chain of infection is that you have this infectious microbe, in this case is a virus, SARS-CoV-2. We have a reservoir or a place where the virus lives, replicates. It'll either be animals, usually different types of animals like bats and so on. We know that there was a mutation that occurred between the bat and perhaps some intermediate hosts that allowed the virus to be able to affect humans and use humans as one of its hopes, right? One of the important things in the infection process is the portal of exit, how the virus attaches and how the virus produces its antigenic proteins or harnesses our machinery to produce them, and then is able to reinfect other cells and then leave the reservoir, i.e. hosts. So in this case, it's usually mouth or nose through coughing, sneezing, or talking. And how do we transmit? Person to person, airborne, direct contact with the virus with a contaminated surface, which is less likely but probable. And of course, the port of entry in the case of the SARS-CoV-2 is basically the membrane, nose, and mouth. That's where the mask wearing comes in. That's why it's so important to block from here to here. For all of you chin mask wearers, you need to pull it up over your nose, right? And what happens at that point is that it searches for other hosts and will infect a susceptible host, start the process all over again. So that's generally the chain of infection. What happened early on in the infection rates, we noticed, is that there were fatality rates that were very high in China. China is where it all started. And we noticed that individuals who had cardiovascular disease, diabetes, chronic respiratory disease, hypertension, cancer were more at risk. In particular, the CD people were about 10.5% at risk for dying if they got COVID-19. So that was very worrying. And what that allowed us to do from getting that data as early as February 11, 2020, is that we were able to put that into our vaccine studies, right? That bit of information. So what we did, and I'm just saying we because, you know, I'm generally belonging to the pool of scientists. I didn't do any of this myself. But what scientists did is that they looked at the way the virus attaches itself to the cells of the body, and in particular the lung alveolar, because that was the cell of choice. And it uses this spike protein, which is this black little umbrella looking thing here, upside down umbrella. And the ACE2 receptor is the receptor of choice, where this spike protein has evolved to attach, enter into the alveolar cell, and then allow that to actually passage through the cell, make copies of itself, and then go on to release its RNA, right? And then the virus now allows the body to produce antigens, repackage it, come out of the cell surface, and have copies of the viruses released on the infected cell. Okay, so the SARS-CoV-2 genome is the genome that we have actually been able to sequence and know which parts.
Okay, we have had a slight interruption in technology, and Dr. Ramlachan would join us shortly. I would take this time to invite you to use the comment field to start typing your questions so that we would have them ready for when Dr. Ramlachan concludes her presentation. So please, I urge you to ask your questions at this time, and I would be sure to post them to Dr. Ramlachan. And Dr. Ramlachan will be getting back in with us shortly. So we stand by for Dr. Ramlachan. In the meantime, I would take this opportunity to urge you to attend our seminars, which would be carded for next week. On May 3rd, Dr. Ramlachan will be sharing on the assessment of risk of the COVID-19 vaccines. On May 5th, which is Wednesday, on next week, she provides insight into whether the vaccine will help long haulers and asymptomatic persons. And finally, Dr. Ramlachan ends this series of discussions on Friday, May 7th, by delving into what is herd immunity and whether it will end the COVID-19 pandemic. All sessions will start at 6 p.m. and can be viewed via the same link used to access this evening's session. So I hope that you would take note of those announcements and you would join us on next week as we continue our discussions on COVID-19 vaccination. So we stand by again for Dr. Ramlachan, who would be rejoining us shortly. At this time, I'd also like to share some information with you regarding the Health Sciences Unit at the University of Trinidad and Tobago and the programs that we have on offer. We have the Masters of Health Administration. We also have our PhD and MPhil courses in health sciences, where we look at equipping health professionals and those wanting to transition into the health field in health training and health administration. So Dr. Ramlachan is back with us and we would resume. Welcome back, Dr. Ramlachan.
Yeah, just don't know what happened there. We'll just resume as much as we can. A little spike, and I believe that is what occurred. Okay, so let's just resume the story. So we go back to the genome of the SARS-CoV-2, right? So I got the little story with the spike protein, the little black upside down umbrella guy, and we were able to actually sequence the SARS-CoV-2 genome very early on. And we took that sequence and we were able to actually generate sequences from them that we can actually use to create vaccines. So how do they do that? Well, there are different forms of vaccines. The first of which that came out were the mRNA vaccines, where they actually packaged the mRNA coding for that same spike protein we're talking about into little lipid kind of molecules and use that as your antigen generating molecule, right, in a vaccine format. There was no infectivity at all. There's no whole virus. There's nothing like that. It's literally just mRNA, a couple of things like sucrose and so on in a lipid droplet. And once that gets into the cell then now can code for that antigen that mRNA codes by, actually produce that antigen, push it out of the cell and present it to its antibody generating cells and say, hi, when you next time you see this, make some antibodies. And that's how you get the immune response that's written, that's written down here, right? So Pfizer, BioNTech, and Moderna uses this mRNA vaccine using the spike protein. It's great in that it gives you a very good immune response without a lot of the dirty adjuvants and those kinds of things that go into vaccines normally. And the memory cells will protect against that particular antigen encountered in the future by producing antibodies to it. One of the negatives about this particular thing, because it's not actually housed in anything else but a fat molecule, it needs minus 70 degrees Celsius because it would degrade very, very quickly at room temperature or anything that's lower. Right, and the cold storage at minus 20 will only last about 30 days for these guys. So you need to have a cold chain that's very effective to be able to distribute this around the country, which countries like the US has. The adenoviral vectors came next. That is the same spike protein coding mRNA, but they put it into a viral vector. And the viral vector is a little more robust, a little stronger, so it can stay outside at room temperature longer. It can stay in minus 20, okay. But the structure is a little bit different. So what it does is that it enters through the vaccine, enters inside of your cell, but then it opens up and releases the mRNA to allow for the encoding of the antigen to be absorbed up by that antibody producing cells and thereby elicit immune response. So it's a little bit different in terms of how it's transmitted, right? AstraZeneca came into being early 2021, and it was approved by the WHO very early on for all adults, including people older than 65, and with the recommendation that it be given in 2 doses, 8 to 12 weeks apart, right? What was great about this is that the SAGE recommendation was right after the COVAX facility, which we'll explain a little bit further on, started to ship it. So that this was the facility that allowed vaccines to reach the low and middle income countries, right? So we got everything kind of happened very quickly and very efficiently at that period of time for the AstraZeneca. The difference with the AstraZeneca is that it uses a chimpanzee's adenovirus, right? So it deletes the adenovirus DNA, so you don't have any disease or anything from that. There's nothing in it that can cause disease. So this idea that you can get sick from the vaccine, as in disease caused by the vaccine, no. It's not infective at all, right? It never goes into the cell's DNA, so it cannot be taken up by your DNA. It is transcribed into mRNA outside of the nucleus, and it goes to the cytoplasm, translates into these antigenic little molecules that mimic the viral spike protein, and then goes out into your body and says, hi, look at me, here I am. Do you like me? Do you not like me? The immune cell says, no, we hate you. We're going to make antibodies. The next time we see you, we're going to attack you. And that's the story, right? What's great about it, it can stay in a normal fridge at 2 to 8 degrees Celsius for a few months, right? No issues. The first injection trials were people were getting meningitis vaccines instead of saline because they wanted to have a realistic effect. So this idea that the trials were not good enough, we don't have enough information, is a little bit, tenuous in fact, because they did actually even more than we usually do for trials like this, right? There was no severe COVID-19 cases or deaths in vaccinated individuals, which was the most important thing. And the blood clots started to be reported in the EU after vaccination. But again, nothing came out of the COVAX facility, specifically AstraZeneca, created in Oxford, right? And what happened with AstraZeneca is that they were able to actually share their patents and be produced in different countries outside of the UK, which was great. So we have AstraZeneca being produced in South Korea and China, sorry, not China, India, and the UK right now, and possibly other countries, right? The DNA viral platform was actually used in completed trials in the US and Brazil. Two-step process as well, 2 vaccines, 1 month apart initially. Now we know closer to 3 to 4 months is a little bit better. And they concluded that it was not only effective against the disease, actually transmitting the disease as well, which was great. And they are for non-for-profit. So they are one of the cheapest, if not the cheapest vaccine on the market at $3 to $5 US per dose, right? And about 3 billion doses have been ordered. The Johnson & Johnson's Janssen pharmaceutical uses the adenovirus that has a spike protein for the SARS-CoV-2 that also has been genetically engineered not to replicate, so it's not infective at all. The strand of the DNA gets into the cytoplasm, goes to the nucleus, and gives the instruction to create this mRNA for the spike protein. Same idea, right? Very similar. It comes out, elicits immune response. The body has memory against that. Next time it sees that antigen in the form of a real viral antigen that comes in naturally, it's going to produce the antibody against that and fight it, right? What is great is 95% of vaccinated people had no severe effects. Nobody died, even in countries with the other variants that was tested in. So Johnson & Johnson AstraZeneca came out later. One of the good things about that is that they already had different variants circulating. So we didn't just have that one single Chinese variant anymore. And we were able to actually test it live to show that it was effective, right? Good thing again about Johnson & Johnson, refrigeration at 2 to 8 degrees Celsius is not a problem. And one of the best things is a single dose, which a lot of people who don't like needles like to hear. It's also non-for-profit, and it's usually priced about $10 per dose, right? What about other vaccines? Well, the Sputnik was the first to come out, Sputnik 5, which was named after the first satellite created on Earth and sent to orbit by the Russians, because of course, they wanted to emphasize that they were the first out. Came out in August 2020. What happened is that they did not do any phase 3 trials. So that's how come they came out first. Their phase 3 trial was donating vaccines to countries and asking them to submit data for their phase 3 trial. So if you took the free vaccines from them, you also would now have to be part of the phase 3 clinical trial, so to speak. So it was a little iffy. A lot of people didn't subscribe to it, but they were able to actually successfully produce this. They claim they have 98% efficacy. And there is a paper that came out February 20th, 2021 in the Lancet, where these individuals, Dr. Jones and Dr. Roy, did some preliminary studies and said it appears to be safe and effective. The Chinese vaccines also came out a little bit later, the Sinovac and the Sinopharm. They're a little bit different in their technologies. The Sinopharm is also an adenovirus, but the Sinovac is a virocell inactivated virus. So what they do is they actually take the actual virus, inactivated, culture it in virocells, and then they use that in the vaccine to be able to create an illicit immune response. A little more tricky doing that, but this is the old school way of how we used to do vaccines quite a bit. So far, they seem to think as well too, they're close to 100%. That's what their reports are. The Chinese are not very forthcoming, I would say, with their data. So it's hard to get realistic data. They also have everybody who has signed up for their vaccines signing non-disclosure agreements where they cannot report on the usage of the Chinese vaccines, which also, again, confounds data and goes against ethical release of information and those kinds of things. So that's the problem with the Chinese vaccine. It's been used and it's been used quite effectively in the UAE and Bahrain and those countries which have been able to reach quite high levels of vaccination using the Chinese vaccines, as well, of course, as China and other areas in Asia. How do they compare? They're similar in that they all use this IDF nucleic acids. There is 2 vaccinations generally, except for the J&J, I should say, needed in 30 days, or as I said, it's been extended now to about 3 to 4 months, anywhere between 2 to 4 months, I should say. It delivers the instructions for the cell to make the protein antigen and stimulate the body to make the immune response. However, the AstraZeneca's DNA, the mRNA is Moderna and Pfizer, and the adenoviral vectors are like Sputnik and AZ and J&J. No need for ultracentrifugation for the AZ. Very high efficacy for half dose versus full dose. And in case of the J&J, it only needs 1 dose, right? There are other technologies that are being used around the world. Canada had some real prospects early on, but then they had to stop because their facilities weren't up to par, and they were doing a little bit more of the DNA based, where they are packaging the same idea, the mRNA, and they're packaging the DNA into the nanoparticles, injecting it via the vaccine or in a nasal spray, which again, a lot of people who don't like needles are excited about. The DNA is released into the cell and it goes into the cell nucleus in this case because it's DNA, and our cells are DNA, so we can use DNA. But it's the same principle. We use the DNA that's coded for the viral proteins, produce the viral proteins, release it into the body to get that immune response and antibody response, right? So 2 exciting things here. You have the nasal spray, and also you would have the lower temperatures, right? And possibly 1 dose if they're able to actually do it effective enough. There are some options as well too from other parts of the world. I think Brazil is doing their own version. I don't know enough about it, but it's supposed to be well onwards in development. They haven't published too much about that yet. Mexico and a couple of other countries in Asia are putting forth candidates. There's a strong candidate in Cuba called Soberana. There are actually 4 versions. The 02, which is the most farthest along in the process in the phase 3 trials, is a conjugate vaccine. So it actually links a weaker antigen with a stronger one. In this case, they use the coronavirus spike protein to a deactivated tetanus toxin, which is very, very strong and should elicit a very strong response. It produces a very powerful antigen that boosts the immune cells and antibodies. And this is a thing of the guy that developed it, Perez Bencomo. That's, that they are still in phase 3. They haven't released it yet. Worldwide vaccines reach per company. Pfizer Biotech is about 72. This is a little bit older data, but this is kind of a breakdown. It's more or less the same. AstraZeneca is a little bit greater now than the Pfizer Biotech. Then the Moderna, the Sinopharm, the Sputnik 5, Sinovac, Janssen, Epivac, Corona, and Covaxin out of India, right? Why is it that you're hearing about the difference in terms of who gets what vaccine and these kinds of things? It really has to take, we have to take a little step back and look at the uptick of the vaccines. This came from Duke Global Health and Innovation Center, and it's an economist paper, and it actually shows you the difference. If you look at Moderna, only available in high-income countries. Pfizer BioNTech, more or less high, upper, middle, and a little bit to the COVAX was allowed, right? And AstraZeneca, again, a lot in the high income, a little bit in the upper middle, low income, but really made up the majority of the COVAX, right? What happened to very early on is that 10 countries bought 75% of the vaccines and have 75% of the vaccines available. And that's the 10 richest countries in the world. If you look at this little correlation graph here, which shows you the coverage versus deaths, the deaths per million at the bottom, and the coverage in terms of confirmed doses purchased is on the right-hand side. If you notice, the UK, US, and Canada had a lot more in terms of confirmed doses. In fact, Canada had almost 5 times in terms of orders. UK had about 3 times. EU and US had about 2 times, 200 times each, right? So they could actually two times. They could actually vaccinate their population twice over. China was a lot less, and a lot of the countries, of course, not even on the scale. So that's what happened a lot to make the situation a little bit unfair a lot early on. So COVAX was developed particularly to kind of get away from that, because they knew that was going to happen, because the countries with money are gonna get the more the greater amount. The countries with access, as in they have the manufacturing facilities in-house, obviously will get more. So COVAX was developed by Gavi, CP, and the World Health Organization, along with UNICEF. And the idea was to develop and manufacture equal amounts of vaccines to guarantee fair and equitable access to every country in the world, right? Obviously, it wouldn't be exactly the same because not every country can afford to pay what the richer countries can. But the intention was to provide vaccines for at least 20% of the population. And with COVID in particular, it's gotten to be about between 2 to 3%, which would cover healthcare workers, basically, right? That's basically what they were saying. And everything else you'd have to kind of buy on your own. But the idea is that you would, they would give these small developing states and the lower population countries a chance to have a small quantity of vaccine at least, right? Which vaccines are in which country? Well, AstraZeneca is the most widely used in 136 countries, Pfizer BioNTech in about 90, Moderna about 42, Sinopharm from out of Beijing 34, Gamaleya, which is the Sputnik 5 in about 30, Sinovac in about 24, Johnson & Johnson about 10, Sinopharm and 2 or 3 countries, and CanSino in about 2 countries. That's the one out of Mexico. The Vector Institute in Russia also produced the Epivac Corona, and that's in basically Russia and Russian related countries, and the Bharat Biotech Covaxin is in India exclusively. In terms of the highest total vaccinations per capita, UK, US, Turkey, Germany, pretty high. India and Indonesia is down here when it looks at the amount that's per 100 people, right? With the highest total vaccinations. Those things have changed a little bit now in terms of populations with at least 1 million people. Israel is #1, followed by the UAE, then Chile, Bahrain, UK, US, Hungary, Qatar, Serbia, Uruguay, right? So people are getting vaccinated in high amounts. This is just a little graphic to kind of show you the difference between the 5 top ones in terms of different characteristics between them, how they made, the doses, storage, and so on. And in terms of the efficacy, how do we know whether or not a vaccine is effective or the efficacy? What does that really mean? So in clinical trials, we measure how well a vaccine prevents illness in a controlled setting, and this is for efficacy. And 95% efficacy does not mean that 95 out of every 100 people will be protected. It means that the vaccine reduces the amount of disease by 95% for vaccinated people. So you're comparing vaccinated people versus unvaccinated people, right? That's basically what that means. And if you look at the variation, now those numbers have changed because the more and more people get vaccinated, the differences that these numbers are going to be also in the population that's vaccinated, right? Pfizer and Moderna early on reported about 95% in disease prevention clinical trials. AstraZeneca and Johnson & Johnson was right around 70%. These numbers are now closer to 87% in AstraZeneca in actual country models, and about 79%, I think, or 78% in Johnson & Johnson. The key thing to note is this line that's highlighted, hospitalization and death prevention. So that means severe disease and death. And what do you all notice? Everything is 100%. So this is really what we want to see. What does that mean? It means every vaccine that we have tested so far has 100% efficacy against hospitalization and death prevention, which is what we're aiming for, right? So they are all successful. More than 75 million people in the UK have received at least 1 dose of coronavirus. They're one of the highest inoculated populations. Number of deaths is less than 10. In the US, it's higher now. It's about 200. Closer to 200 million have received at least 1 dose. And the number of deaths a little bit unknown in terms of that are specifically to the vaccine without risk, but they're in roughly about the 10s or 20s, right? After the EU and everybody did their pause on certain vaccines, they've now raised their pause and said that, you know, it has to be evaluated on the basis of risk in a per case basis, and it's okay to use based on their data, right? For AstraZeneca and J&J. In Israel, just about 16 out of 700,000 people were hospitalized with COVID-19, so it's very effective. Nobody died as far as they are known in terms of SARS-CoV-2 vaccination in the COVAX facility delivered to date around the world. Vaccines are not effective. People keep saying that. AstraZeneca only has 66. Well, as we just discussed, it's a little bit higher, but then 66 is still very high. If you look at the seasonal flu vaccines, we usually only get about 30, and we jump for joy if we get 30. Anything that's around 30 is considered to be a great success when it comes to influenza vaccines. The most important thing is that they are 100% effective against hospitalization and severe disease and death, right? We talked about the variant situations last week, and I just want to tie that back into the vaccines again today. Well, not last week, last Wednesday, I should say. And they both appear to be the variants that are popular, the Kent and the South African and the Brazilian. They seem to be able to be effectively prevented against infection with the AstraZeneca and the Sinopharm, which are the two. The Sinopharm's a little bit lower, AstraZeneca's a little bit better. If we look at corona vaccinations, as of today, April 29th, 2021, yesterday's data, which is available today, just about 1 billion vaccines have been administered, which equals to about 14 doses for every 100 people. There's already a very stark gap based on the countries, as I discussed, with some yet to report a single dose, right? So Barbados is right about 73,590 in our region, about 26 per 100. Guyana is about 15 per 100. Trinidad and Tobago is right about 2%, 2 per 100 at around 697. This is a little bit higher now today. This data was downloaded today, but it's a little bit higher. We've had 2,000 donated by Barbados in January, February. 33,600 doses arrived on March 31st. That's the first COVAX delivery. It's going to be done in 2 tranches. The second one is going to come before the end of May. 40,000 doses from India directly from the SSI, which is producing the Covishield version of the AstraZeneca, and up to 100,800 on the COVAX order. We are just at about 2%, and we really need to get closer to the 90-something%. I wanted to show you all the relative, this little graph, which I thought was really powerful. So this is COVID-19 vaccine doses administered per 100 people. Bermuda, which is a very small country, was relatively very easy to vaccinate very fast. So they're already in the 90%, right, of vaccine doses. So they're actually close to their population already getting their second doses. US has also been doing really well. They are not there yet, but they're almost there. I think they're about 65 completely vaccinated and about 80 something, at least 1 dose. Canada started off very slow, but it's been creeping up. But look at our region. Dominica, Antigua, Barbados doing very well. Guyana, St. Lucia, Grenada, trying to reach about 20%. Jamaica has fallen behind. It started off pretty good, but they've fallen behind now. And Trinidad and Tobago is really at the bottom in the region for vaccines administered per 100 people as of yesterday's data. Israel is the highest at about 121. Then followed by the UAE, which I mentioned, that got a lot of the Chinese vaccines. Israel got a lot of Pfizer, and they got a lot of Pfizer free because, again, they were involved in the phase 3 trials. That was part of the deal. Same thing with the UAE and the Chinese vaccine, and Chile with the Chinese vaccine as well, and Bahrain with the Chinese vaccine. US, UK, Hungary, Serbia, and then we go all the way down here. India is pretty badly off right now at only 10.82%. A lot of people feel that they're making vaccines, so that means they should have vaccines for their people, but that's not how it works, because the SSI, even though it's the largest manufacturer of vaccines in the world, have been contracted to patently produce the AstraZeneca for other countries, not for India. So they're behind in terms of their vaccination schedule. And you have to remember, if you look at their number of doses, they're probably second in the world for the number of doses administered, because they have 1 billion people. They have only given about 10% of that. And Trinidad is right down here, roughly at 3 on this calculation, right? What's the future? We have to understand there's still hundreds of thousands of people getting sick. This has not gone away. In fact, everybody's on uptick. If you all remembered from the last lecture, all the graphs are ending up in a peak right now. The only graphs that are decreasing are the individual countries like Israel, US, UK to a lesser extent, but now it is getting there. Bahrain, where vaccination percentages are so high, we're starting to see the effects of herd immunity coming into play and the numbers of new cases dropping, right? We cannot depend on one type of vaccine to achieve herd immunity. All the types have to be available to get population access. International flights, schools, public and private service, they all will depend on proper vaccination. And until it's widespread, we're not going to be able to have that response and be able to control the population, the pandemic. We still have to continue to practice physical distance, hygiene, all of that, use of masks in particular, adequate ventilation, avoidance of crowded spaces in the foreseeable future. There are people putting forward suggestions to stop wearing masks if you're vaccinated, if you're out in the open areas, if you're with your family groups, those kinds of things. I would advise against that so early on in the vaccination process. We really have to get to a point where we have good herd immunization going on. And of course, herd immunity, which we keep talking about in every lecture in this series, is when we're going to get a high enough people to be able to protect the other people. And then the virus is harder to spread. It does not have a host anymore to pool and mutate and become stronger variants. And basically, we see those numbers shrinking. And that really would only happen through immunization, right? Tune in on Monday. We're going to go into the COVID-19 vaccine safe story. And I'm going to be kind of doing a little bit of the vaccine mythbusters, right? The New York Times just released this yesterday. No other people's COVID vaccines cannot disrupt your menstrual cycle, because a lot of these websites are reporting all kinds of crazy things. And I kudos to the media and the New York Times for responding in an area like this, so that we don't, as scientists, have to now come up with a reason why this doesn't happen. They're actually telling you, it's not possible to experience any effects from being near to a vaccinated person. That's a key point, because the vaccine ingredients are not going to jump out of their body and jump into you and affect you in any way, right? So this is just a little teaser for our next episode. So tune in, and thanks for joining me.
Thank you, Dr. Ramlacha, and I look forward to next week's presentation, where you would debunk many of those myths which would have contributed to a lot of hesitancy in taking or considering taking the vaccine. At this point in time, we would now turn to the question and answer segments, and I'm seeing questions coming in. So we'd like to start with a question from Kirian, and Kirian is asking Dr. Ramlacha, what are the chances of new mutations occurring, leading to new strains of the virus in our country? And should we be prepared for a scenario like that?
Well, Kieran, I would hope that you would have gotten that answer from my last lecture, which you attended, on the variants and how easy it is for the virus to mutate once it has a host, right? The point is that you have to reduce infection. Once there's a pool, i.e. unvaccinated individuals in a population that act as hosts, these viruses are going to mutate, and more variants are going to come out. That's just the nature of viruses, right? So yes, mutations are going to happen. There is now a double mutant in India that they suspect is causing a lot of the deaths that they're seeing. Their rates are very, very high. I discussed that a little bit on Wednesday, the last lecture as well. And that is literally because there are so many people who are still unprotected, and there's a host that's going to allow that to happen. Many hosts, I should say. So as long as that is occurring, there's going to be a chance of different mutations occurring. Right now, in terms of the variants of high consequence, we don't have any. This one in India might be. It hasn't been documented to be yet. It's still a variant of concern. And that means it possibly can evade immunity. It possibly can stop the efficacy of the vaccine. It possibly can cause increased severity of disease and reduce immune response. So all of that is still possible. So to answer your question, yes.
Thank you. Dr. Varma Rambaran is making the comment. The Honorable Prime Minister announced yesterday that the booster shots, which were reserved for the first group of vaccinated citizens, would now be consumed as first shots for more citizens. Noting that AstraZeneca is to be taking as 2 doses, what will be the fate of the patients who took only the first shot alone?
Well, that is a distribution decision that has to do with the fact that there is a life for the vaccine. The vaccine expires at a certain point in time. So that's 40,000 that came from India last month is not going to be good for very much longer than 3 months or so. The individuals who got vaccinated in April were told to come back in 3 months. So that would be July, right, for their second shots. So there's no reason why he should not use the, in my opinion, the vaccines we already have here to increase the single dose percentage in the population. and with the assumption that by June or July, we're going to get the rest of the COVAX and other facilities being able to deliver and meet the second dose of those individuals. And then 2 to 3 months later from May, have the second dose for the individuals who are getting in May. So I think that is actually a very effective strategy. It makes no sense to have those doses sitting down in the fridge and they're individuals who are not vaccinated.
Yes, and I would just like to ask this question. Are there any implications regarding changing the period of time between the first and the second doses? Because I know that in some cases, persons would have been given, perhaps in the UK, they would have been given a shorter time to come back for their second dose, and then it would have changed due to shortages. So they would have gone from perhaps 6 weeks to about 3 months, 2 to 3 months after. Are there any implications when you change that period?
It wasn't really because of shortages. It really was because they were noticing that actually we're getting a better antibody response if you waited a little bit longer. Initially, if we go back to June, July, August 2020, everybody was in a rush for information. So when they were doing their phase 1, phase 2, and eventually their phase 3 trials, they shortened the period of time to be able to give the second dose, to be able to get that data to say, yes, okay, well, our vaccine working, give us the authorization and let's go. So we didn't have the luxury of waiting 3 and 4 months to see whether or not the antibody response was better. In fact, they thought, well, okay, well, if the antibody was so good and we got 95%, and in the case of Moderna and Pfizer, so early on, and AstraZeneca and J&J, in the 70s, 60s, and 70%, let's just go with that. J&J, of course, is a single dose, and they're okay with that. But the efficacy is quite good with that as well. The underlying reason for that is because they didn't have the time. Now that we've been able to have the data, we can say, okay, effectively, yeah, if you wait because there are a lot of people who are missing their dose, and there's a lot of information that's we're gathering, right? So even though some people were given exactly 30 days, they came back later, and then they started to realize, well, there's no difference in response. Let's use these first doses to vaccinate more people, and then now give a bigger window to have a better antibody response, which is second dose later on. So it's all working parts and parcel. And a lot of people have to realize the reason why recommendations change. It's not because anybody's having a conspiracy and, Bill Gates wants to give 5G in your vaccine and, barcode you or anything like that. It's because we are in the process of gathering data. And as data comes out, the recommendations are going to change, rightly so.
Okay, good. Kirian is asking Dr. Ramlachan, it seems that China is experimenting with giving different vaccines if multiple doses are required. They're essentially mixing vaccine, and he wants to know your thoughts on mixing vaccines.
Their vaccines are different technologies, right? So their vaccines are, as I mentioned, the old school vaccines that we have where we take deactivated virus or attenuated virus or change virus and grow it up in cells and then extract that and use that as your antigen boosting part of your vaccine, right? So they are able to mix their technologies just because they make the beginning starting point is exactly the same. So a lot of their technologies are similar, so they can mix vaccines in terms of what they are producing. So I don't see a personal issue with that. It's just I wish they would publish their data a little more effectively and get the word out there, because if it is that it is effective, and I mean, I know personally the UAE royal family got the Sinopharm, and a bunch of people, as I mentioned, Sinopharm is utilized quite widely across the Middle East and quite effectively. So, I mean, the data is there. They just, being a little secretive with it. So, as more and more data comes out, I mean, there's a couple of publications that have come out and said, yes, it's very effective. And that strategy might be a useful one, because then it will allow manufacturing facilities to be able to catch up and be able to give production for other parts of the countries that are behind.
Which individuals should not be given these vaccines? In other words, are there any health conditions that make individuals ineligible for vaccination?
Right. So we'll talk about that a little bit tomorrow. Not tomorrow, I keep saying tomorrow, Monday.
Monday.
Yeah, in terms of are they safe? There's a strict instruction. If you are allergic to vaccines, if you have gotten allergic vaccine in the past, to a vaccine in the past, you do not take it, right? That is just standard because you know you are allergic. If you have to carry on an EpiPen for those kinds of things, you need to speak with your doctor specifically, go through each ingredient in the vaccine that you are going to take and determine whether or not any of those ingredients have caused allergies in you in the past that would create some sort of anaphylactic shock, right? Right now, it's approved for breastfeeding. Certain vaccines are approved for breastfeeding and pregnant women. I believe the Pfizer has, again, I'll talk about that on Monday specifically. And the Pfizer is also approved for 12 to 15-year-olds, I believe. Now, they haven't EU approved it yet, but they've just recently released studies that show that it's safe in those populations. In terms of Trinidad and the AstraZeneca, which is what we have available, and our region in the Caribbean, which is mostly AstraZeneca, a little bit of the Chinese, but mostly AstraZeneca, the clotting issue is a problem. So individuals who have a tendency to clot know they have thrombolytic kind of thrombolytic kind of situations, or people who have thrombophilias, those kinds of things. There's a genetic test you can do that's done here locally at Genetics Diagnostics, which I'm affiliated with. There are different tests that you can do to determine your risk that way. If you are prone to strokes, if you are prone to anything like that where clot forming, if you've had DVTs in the past, those individuals will have to have discussions with their doctors whether or not they are prone at this particular time. Do blood tests, check your platelet levels. Don't do these things blindly. Nobody's asking you to do blindly walk in and take whatever is offered. That's not the point of the story. The point of the story is find out about your own personal risk. There are tests you can do. You can have discussions with your doctors to determine what is your status, whether or not you can take it. In terms of individuals who've had COVID-19, it's recommended that they wait at least 2 weeks. If you want to know if you've been asymptomatically exposed, you can do a COVID-19 antibody test, which we do as well. Agenics and other labs do and other people do to determine exactly what is your level. If you have a strong antibody response, it's recommended, again, you wait 2 to 3 weeks to make sure things kind of calm down, because that's one of the reasons why you might have counter indications with taking the vaccine.
Yes, and I think that you would have touched on this last question being asked, what are your thoughts on the vaccinations for teens and kids? So I'm hearing that there's still a need for more data as it relates to kids particularly. I know that you did mention 12-year-olds.
Well, they're going to, what happened is, as I mentioned, they took the most susceptible population first into the initial phase 3 trials, which would be the obesity, diabetes, cardiovascular risk, that group, in addition to, of course, normal people who have no comorbidities. And they tested them to make sure, because they were the ones who would drop in dead very, very quickly. And the fatality rate was very, very high. So those individuals got checked first, and we know it's effective, it's safe, and the recommendations came out first. The next group would have been the pregnant and breastfeeding, which was just started to be published. The other groups are the 12 to 15 or 12 to 18, or 12 to 15, I think, because 16 to 18 was included in the adults. And that's coming out soon. And then we're going to do the next set, which is the under 12. And then, of course, the infants, which would be the last group. Just basically based on infectivity and susceptibility, nothing more than that. The new variants in Brazil and India seem to be affecting younger individuals. So it's become a little more urgent for us to get that data out. But there are individuals that have been working on that as well too. But eventually, 70 to 80% of the population of the world that can take the vaccine are going to have to take it for us to be able to get the effective herd immunity. And it will eventually go to a point of having to probably get boosters or those kinds of things. Pfizer's already said that they want a third booster attached to their vaccination protocol. That might be something that might come up, but I mean, it's not that much different from influenza, where we have to get a new vaccine every season. So that could happen with teens and children as well too. That it'll be included in part of the vaccination protocol at birth. So their first set of vaccines at 3 months, 6 months, boosters at 3, 4, 6 years, whatever, would be included. So
okay. Yes. Sorry?
No, I just said thanks for that question. That was a good one.
Yes, and that concludes our questions. On behalf of the Office of the Vice President of Research, Academic and Student Affairs, the President of the UTT, Professor Prakash Prasad, and our Board of Governors, I wish to thank you, Dr. Nicole Ramlachan, for taking the time to educate and inform us about the effectiveness of COVID-19 vaccines, and more so the importance of being protected. I also wish to thank our viewing audience for their participation and to encourage you to join Dr. Ramlachan in the upcoming webinars. And so until then, I